The cytolethal distending toxin is present in gram-negative organisms such as Haemophilus ducreyi, which causes the sexually transmitted disease cancord. The cytolethal distending toxin is composed of 3 parts, which consist of a catalytic subunit CDTB and 2 cell binding subunits CDTA and CDTC. The cytolethal distending toxin will bind to the lipid rafts of the plasma membrane of a cell and is internalized by endocytosis. Through the process of endocytosis, the cytolethal distending toxin will travel to the early and late endosomes, where CDTA will dissociate from the toxin. It was hypothesized in our experiment that acid-induced changes to a CDT subunit is responsible for CDTA dissociation in the endosomes. Variations in acidity within the early and late endosomes may affect dissociation of CDTA from the CDT complex and structural changes to the CDT subunits.
Moving on to the results, we confirmed that our proteins were pure as shown in our SDS page gel in Figure 2, Image A, and we ensured that our CDT proteins were also functional using an MTS cell viability assay as seen in Image B. In addition to studying how acidity impacts the dissociation of the CDT holotoxin, we also used circular dichroism to analyze changes to the structure of our proteins. Referring to our results in Figure 3, we found that at pH 5.2 and 6.3, CDTA was the only subunit to experience structural changes. We were also able to demonstrate CDTA precipitating out of solution at acidic pH in Figure 4. Precipitation at acidic pH did not occur in CDTB or CDTC, once again confirming that CDTA is the only subunit significantly impacted by acidic pH.
As seen on Figure 5, side secretion chromatography determined dissociation of the holotoxin under acidic conditions. At pH 7.4, all subunits were found at peak 0.36, and at 0.58 peak, only CDTB and CDTC subunits were found. At pH 5.2, no holotoxin was found at the first peak, but the 0.58 peak remained unchanged, suggesting precipitation of CDTA out of solution. All of this was confirmed by a Western blot. We can conclude saying this assembly of CDT is needed for proper desiccation of the cell. Our results reveal changes in the structure of CDTA causing dissociation of the holotoxin. Future studies are focused on structural changes at pH 6.3 for better understanding and production of therapeutic treatment.